Good morning and good afternoon to all of you for joining our lab channelopathies. My name is Jason Gomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's event alongside COO of Nanion Technologies North America, Rodolfo Aedo. Today's speakers will be Professor Al George and Jen Pan.
Before commencing, we'd like to make a quick introduction about our speakers today and their respective presentations. For a brief background, Dr. Al George has utilized the SynchroPatch for over 6 years now and has a 768 format, which equates to having 2 3D4 modules inserted into one liquid handler, yielding 768 recordings in parallel. And Dr. Jen Pan purchased the SynchroPatch shortly after in 2015 with the single 384 format or one module embedded in the Beckman liquid handler. Together, they are recognized as early adopters and were the first 2 core facilities in the US to utilize the SynchroPatch to characterize hundreds of different functional variants, both loss of function and gain of function, associated with neurodevelopmental disorders such as autism, epilepsy, and schizophrenia. The talks will be primarily focused on the channelopathy theme, highlighting respective projects that Al George and Jen Pan have been working on in their labs. Over the course of this presentation, you will learn about their research initiatives and why they're both regarded as pioneers in the field of ion channel research. Thanks to their work, they've been able to correlate various clinical phenotypes with ion channel dysfunction directly in a systematic and high throughput approach.
Today's presentation are as follows. Professor Al George will discuss his work in decrypting variants of unknown significance in the channelopathy-associated epilepsy. Al George serves as an adjunct professor and chair, Department of Pharmacology at the University of Northwestern, Feinberg School of Medicine. His lab was the first to elucidate the functional consequences of an ion channel mutation linked to an inherited cardiac arrhythmia, long QT syndrome. This discovery contributed greatly to understanding arrhythmia susceptibility into disease and inspired use of drug targeting persistent sodium current as a therapeutic strategy. Jen Pan will discuss analyzing rare variants of CAC-NA1I, which is a type T voltage-gated calcium channel, also commonly called CAV 3.3, derived from schizophrenia patients. Jen Pan is the Director of Translational Neurobiology at the Stanley Center of Psychiatric Research at the Broad Institute of MIT Harvard. The research of her group focuses on translating emerging genetic into biology and to enable next-generation therapeutics to treat psychiatric illnesses. Upon completion of the presentation, we will hold the Q&A session and as such welcome you to ask the presenters questions at any time throughout the presentation. You can log a question via the chat window found on the right-hand side of your screen. If you happen to experience any technical difficulties at any time, please inform us via the chat box. Today's presentation will additionally be made available on demand shortly upon completion of the event, inclusive of a transcribed copy of the Q&A. I will now turn over the presentation to Jen for the start of the talk.
Okay. Hello, everybody. This is Jen. I'm delighted to join Al to give an introduction to the work that we do. And so we discussed a little bit, and I thought that I could start this discussion more on the technical side and talking about the automated patching using SynchroPatch 384 patch engine. So here's an outline of what I'm going to talk about. I will give a brief introduction of what SynchroPatch do, and what is a typical process of the experiments and the type of experiments that we do using SynchroPatch. And then I will give 2 examples of my group. One is a ligand-gated channels. I thought I'd profile a little bit of the diversity of the assays that we could use SynchroPatch for. And then I will focus on the CAV 3.3 and in particular I wanted to tell you about this noise analysis that we were able to utilize SynchroPatch for, which is typically very tedious, using the manual patch. And then I'll hand it over to Al for him to continue the discussion. So many of you are very familiar with patch clamp technique. I just wanted to highlight the difference between the manual patch and the auto patch. Can you see my cursor? No? Okay. So typically for the manual patch clamp, the electrode is approaching from above the cell, and then you form a seal, and then you break open to form the whole cell configuration. Perhaps configuration-wise, perhaps the distinctive feature of the planar patch is that the cells are sitting on top of an aperture, and that forming the whole cell patch configuration is from the bottom. So I have, in my experience, I've used 3 different types of automated patch, planar patch. The first one that I used is the Ironworks, and then subsequently I also have used the Patch Express from Molecular Device, and currently at Broad, what we have is a SynchroPatch, and then I have SynchroPatch. And here is just a brief comparison of the different systems. They all each have their own features. Ironworks is great for as an engine for pharmacology and screening. The one thing to understand about the Ironworks is that it's where the population patch clamp started, and it's a mega-ohm seal, versus the Patch Express and SynchroPatch that offers a giga-ohm seal. And their fluidics is a little different too. As shown on the rightest hand, the SynchroPatch doesn't have individual fluidics, but is using the pipette as the main addition and subtraction. So when we started working with SynchroPatch, this was funded by Broad Next 10 to my group, IST, at Broad Institute. We purchased the SynchroPatch 384 back in 1915, and we mainly use this instrument for 2 types of analysis. The first analysis is the large-scale variant analysis. We, at Broad, we have identified many coding variants as potentially associated with disease risk, and the manual patch is just not at the scale where we could draw any conclusion from this large genetic studies. And then so the automated throughput electrophysiology is the only way to get down to the bottom of translating human genetic findings into potentially molecular insights onto how the risk is associated with individual ion channels. The second type of experiment that we use SynchroPatch for is the throughput pharmacology. This perhaps is more common for the industry users, where this is a very typical pharmacology experiment using many different compounds and look at either at the single dose or at the dose response. We have done mostly with the dose response and use the SynchroPatch as a secondary assay, not the primary assay for the throughput screening. So this is exactly where our SynchroPatch sits in the lab. It has been with us for several years, and it's really our working horse engine for many of our scientific inquiries. I wanted to give you a quick sense of what is the workflow working with SynchroPatch. So it basically divided into the system setup and then the interrogation into specific aspect of the function. So the setup includes the mechanical side. You need to fill the chip and you add the cells to the plates, and then you need to wash and form the patch, adjust the amplifier, and then have the configuration correct and pass all the QCs, and then you're ready to record. And then, as I mentioned, then the next phase is divided in our mind into 2 types of experiment. One type is the phenotyping, and in our mind, it's typically analyzing a large set of coding variants of ion channels. And then the second type is pharmacology. And then for the phenotyping, we do a QC run first using the test pulse, and then we apply the voltage protocol 1, 2, 3 potentially, and then between the protocols we use solution exchange to maintain the solution. And then for the pharmacology, typically it's analyzed before and after the compound addition. So here is just gave you a quick sense of what the synchropatch assay voltage protocol look like. And for those of you who are familiar with ClamFit, you can see this is rather similar to a typical voltage setup in either ClamFit or other forms of the manual patch software. You can specify as many voltage steps as you want. It's just really comparable to what a manual patch is able to do. I think where when we first started working with Synchro, we totally underestimated the amount of the data coming out from synchropatch and how powerful this engine is. Once you record, it's 384 wells, and then if you have 80% success rate, you end up with 300 individual cells, and the analysis is a bear. So to that end, we worked with both NanIon as well as we developed our own software package to both QC as well as analyze and compare different variants across different variants and before and after the drug addition. I think that's really a part that we learned as we go. Both the data generation using synchropatch is not a bottleneck anymore. Compared with manual patch, the throughput is there, but how you look at the data, the analysis side is really what allowed us to be able to dig more into the biology. And then we also spend a lot of time to visualize the data. What makes most sense to look at what aspect of the properties of the ion channel. I won't go into it. I'll show you some examples at the end of my talk, but just to say that given this much of the multidimensional data that we collected per cell, per variant, to be able to get a holistic feel and understanding of what each variant do or what drug does, really is a big part of the challenge using the synchropatch. Now that data generation is not a bottleneck anymore, how we visualize this data, how we take in this part of the data is a big part of our consideration currently. So with that introduction, I'm going to give you two sample studies that my group has undertaken. The first sample is actually a small project that I collaborated with Weifeng Xu at the time. She was at MIT, and we looked at NMDA receptors and variants of NMDA receptors. And then just to give you a little bit of background, so this is Weifeng's project, and then she looked at the neurogranin, which is the risk gene for psychiatric illness. Psychiatric illness is my primary focus, and Weifeng and I discussed about this. What she discovered is the neurogranin is a calmodulin regulator, and then she believes that neurogranin knockdown alters, basically blocks the potentiation of the plasticity, synaptic plasticity. And then she did proteomics and isolated the green2A has phosphorylation sites that is induced by neurogranin knockdown. So with all of this work that Weifeng did, and she came to us and then said, well, can you do something about the C-terminus of phosphorylation site? So what we did with WeFun is that we constructed 4 cell lines, okay? So we generated NMDA receptor cell lines that are composed of 2 obligatory NR1 subunit and then 2 optional NR2A subunit that is encoded by GRIN2A. And then they are linked by a P2A site so that we ensure that at least at the mRNA level, they are consistently expressed. So on the NR2A side, we have 4 different variations. So one variation is the full length, the wild type with this 4 different sites that WeFun has identified being phosphorylated. And then we have the NR2A that missing the C terminus. And then she further made a phospho-mimic and phospho-dead, where the serine is changed into all aniline or all the lysine amino acid. We validated all of these cell lines and we developed assays. I do wanted to show you some of the assay development that we did. And so initially we did the experiment on the left side where we had the recorder baseline at the ligand and then had a washout. And then on the right side, we did a much longer time pause after we add the addition using this stacked ligand addition. So basically the ligand is only added on the tip of the adding pipette. And then so you cannot really precisely control the ligand addition at the millisecond level, but you get a proximal readout of their deactivation or desensitization. So with this assay in mind, at our hand, we were able to look at the 4 different variants, and 3 different variants compare with what wild type NMDA receptor would do. And basically, we were very happy to see, if you look at the B first and only the wild type, that there is a calcium-induced inactivation of NMDA receptor current that is largely removed in the presence of the barium. And on top of that, the C terminus is, if you don't have the C terminus, that this calcium-induced inactivation seems to be gone. And we further see that SA, the phospho-dead and phospho-mimic, both have some sort of deficit. We couldn't explain why SA and SD perform same exactly. WeFun had some hypothesis there, and we're following up with more biology. But I just, this work is impressed. And then what I wanted to drive home is that because of this assay we did, and then because of the numbers that we were able to do with the help of SynchroPatch, we were able to perform this experiment rather quickly once we have the cell line, and it enables us to really analyze the C terminal truncation and phospho-dead and phospho-mimic in a very short period of time with quite a good control on the activation side, and we indeed can read a deactivation as well. It's not as precisely controlled as a manual patch with a ligand, fast ligand-gated channels. However, because of the numbers we were able to get, we can have a precise, at least a very consistent readout of how they behave in the setting that we have, the protocol we have set out. So in the next few minutes, I'm going to switch the topic and give you another study example of voltage-gated channels. So we covered the ligand-gated channels in the last example using the SynchroPatch recording. And so this next example has something to do with voltage-gated calcium channels. And my work is mainly focused on the psychiatric, the genes associated with psychiatric risk, and then how we can find potential therapeutics, novel therapeutics for severe psychiatric illness such as schizophrenia. And one of I'll skip all the background, we don't have time for that, but all what I wanted to say in this slide is it just happened so that there is one gene that is implicated in the risk of schizophrenia, and that is the CACNA1I. And just give you some reference point, the dopamine receptor 2, which is the target for all of the antipsychotics nowadays, is also a hit in this GWAS Manhattan plot. So we knew that this was a risk gene for schizophrenia, and we were interested in studying it. KCNQ1I is a novel target for schizophrenia, encodes a voltage-gated calcium channel, and with all the genetics, all the biology behind it, we worked with a genetics group here at Broad, and they have a complete Swedish cohort, about 10,000 people. Approximately half of them are schizophrenia and half of them are from control patients. So from that, we identified about 23 ultra-rare variants. Ultra-rare variants means that these variants are present in the population in less than 1 in 100,000 people. So the reason we're interested in ultra-rare variants because that in the genetic analysis, ultra-rare variants seems to confirm most of the risk in schizophrenia patients. So at that point, we set out to analyze these 23 ultra-rare variants together with 7 common variants that we are able to identify. All of these are coding variants, of course, in this Swedish cohort. So how do we do that? We did it using SynchroPatch. So this is on the left side, it's a plate map. You can see it's 384 wells, 16 times 24. And then what's shown in green here is the CL resistance is more than 1 gigohm. And then the blue ones are more than 500 megohm. So, I mean, this is one of the better examples, but in the gist that we have a very good luck with this particular cell line that gives us a very good success rate on SynchroPatch. And then we used 4 different parameters to characterize this particular calcium channel gene that includes voltage dependence of activation, voltage dependence of inactivation, recovery from inactivation, and current density, which is core property of ion channels for conduction. We did a quick comparison on one of the variants between the automated patch versus the manual patch. The absolute V-half may be different because the solution we use is slightly different, but the shift of the variant with regard to the wild type seems consistent using manual patch versus SynchroPatch. And then with that in mind, with the validation in mind, we went ahead and did the characterization of the 30 variants, including 23 ultra-rare variants and 7 common variants. And then we analyzed the properties in 4 different domains: in the activation domain, inactivation domain, peak current density, and recovery time. So what we discovered is perhaps is better summarized in the next slide, where each corner represents one property of the channel. And you can see that the ultra-rare variants derived from schizophrenia are dominantly pointing to most of the variants change the properties in the north corner, while the common variants didn't change much of any properties. And unaffected mostly focus on the west corner. So we thought we thought that this is very interesting, and only by analyzing a large set of variants, and you'll hear from Professor L. George in the next section, that only by analyzing a large set of variants that you start to have an insight on the molecular phenotypes that potentially be associated with disease risk. So, and then this won't be possible by manual patch and can only be achieved by instrumentation like a SynchroPatch that can analyze a large set of variants with a lot of numbers. So I will skip through this. I just wanted to have I'm going to go directly to this technical point, is that we used SynchroPatch to do the noise analysis. Typically, it takes, the manual patch is tedious and it's quite hard to do. But with the numbers that we get from Synchro, we were able to do a noise analysis on most of the variants that we have and estimate both the number of channels on the surface as well as their unitary conductance in the physiological conditions for many of our variants that's presented here. And then we have, in the end, we identified a couple of variants that are changing either the conductance or the amount of the surface, the amount of channel that can traffic to the surface using this noise analysis. I just wanted to close by acknowledging my team. This is ICE-T at Broad, Ion Channel Electrophysiology and Technology. That most of this work on the NMDA receptors are carried out by Andrew, who's on the left backside, and then the work on the CFD 3.3 is carried out by David Baez, who's in a red T-shirt and in a back row. And I have a fantastic crew to support me through this effort, and for Broad for funding us and with their continued support. With that, I'm going to hand it over to Professor Al George.
Thank you very much, Jen, and thank you to Nanion for hosting the webinar today, and thank you for people who've tuned in to listen to our presentations. So I'm going to tell you one of the many applications of the Synchro Patch that we've enjoyed over the last 5.5 years or so. This is a story about epilepsy and our attempts to understand the functional consequences of genetic variants in just one of the many ion channel genes that are associated with different forms of childhood onset epilepsy. And that gene in particular is KCNQ2. So if those of you who are not familiar, epilepsy is a very common neurological disease affecting approximately 1% of the population worldwide. Many people have unclear etiologies of epilepsy, and what used to be called idiopathic epilepsy is now referred to as genetic epilepsy, because there's lots of data now supporting the idea that epilepsy is a complex genetic trait in most individuals, but a very small fraction of cases is the result of monogenic forms of epilepsy, including many forms that are associated with ion channel genes. This slide illustrates just some, probably most, but not all, of the ion channels that are associated with epilepsy. And this includes several voltage-gated sodium channels, voltage-gated potassium channels, calcium channels, as you just heard from Jen, and ligand-gated receptors. And these, in addition to this genetic heterogeneity, there's tremendous phenotypic heterogeneity. There's several well-defined syndromes that are associated with ion channelopathies. And I'm not going to dwell on the phenotypes per se, but I do want to emphasize that among the ion channel genes associated with epilepsy, there's also tremendous allelic heterogeneity. And this slide just illustrates the number of allelic variants that are found in different ion channel genes in epilepsy. With SCN1A, voltage-gated sodium channel NAV1.1, being the gene with the highest number of variants in the disease. Other sodium channels and potassium channels dominate this list. So this idea of allelic heterogeneity is really what kind of got our attention years ago, both in the brain and also cardiac channelopathies, and trying to understand the functional consequences of these genes one at a time with manual patch clamp became completely impractical. Now, one of the important aspects of knowing that there are lots of genetic variants being discovered is the classification of those variants by geneticists. And genetic testing laboratories in particular typically use a scheme such as shown on this slide, where the variants are classified based on the probability that they are the cause of the disease. And when the evidence supports a high level of confidence that a variant is the cause of the disease, the variant is classified as pathogenic. When there's less evidence but still supporting pathogenicity, a variant may be classified as likely pathogenic. And then on the other end of the spectrum are variants that are likely to have no impact on the disease, causality, and those are benign or likely benign. Unfortunately, in the middle is a category called variant of unknown significance. And this, unfortunately, is one of the largest categories for many genes. For KCNQ1, it probably represents about a third of the variants that are deposited in the ClinVar database. And so this represents a very large chunk. And any variant of unknown significance really is not diagnostically valuable in many cases. And so one of our ultimate goals is to be able to perform functional studies that may offer additional evidence supporting either pathogenicity or the lack thereof. And this is where the SynchroPatch has come in and transformed what we have been trying to do. So this is the picture of the SynchroPatch 768 patch engine that's in our laboratory. It's just a blow-up of the 384 well patch plate and a little schematic here showing the laser-engraved hole that's present in the bottom of each well to form the planar patch electrode, as illustrated in the cartoon. And this system is really very powerful. It has allowed us to go from studying just a few variants at a time to literally dozens of variants in a week. The machine is very sophisticated, as you've heard from Jen's talk. It's so sophisticated, it has to have its own Twitter account. So just so you know. So our approach is a little different than the approach of our colleagues at the Broad in that we don't make stable cell lines of each variant. Early on, we thought about doing that, and a member of my laboratory suggested we try a form of electroporation that could give high transfection efficiencies. And so we have optimized this system called the MaxCyte STX, which is a high-end electroporation device widely used in industry. And we've been able to couple that with the SynchroPatch to avoid the need for making stable cell lines, and we've been very happy with that. The scheme is usually, what we have typically done is we create our plasmid vectors in a vector backbone that has an IRES element followed by a fluorescent protein. And in the case of KCNQ2, which is the highlight of my talk today, we have to combine that with KCNQ3, which is an obligatory co-subunit to reconstitute the neuronal muscarinic modulated current called the M current. So in the beginning, we actually co-transfected KCNQ2 and KCNQ3 together in plasmid backbones that had either GFP or a red protein called M scarlet, transfected them into CHO cells, and then used flow cytometry to quantify the transfection efficiency of both components. And as illustrated in these flowgrams, we were able to get upwards of 90-some% transfection efficiency of KCNQ2 using the GFP fluorescence as its proxy, and similarly close to 90% with KCNQ3. These are not atypical experiments, and so we felt confident that we could conduct reasonable experiments. And so this is a typical experiment on the SynchroPatch for the use of co-transfection of CHO cells with KCNQ2 and Q3. What you see here are these beautiful, robust outward currents that resemble the kinetics of the M current, a few exceptions here. We get a large fraction of the cells to have a gigaohm seal. In this case, I think it's around 310 cells from this experiment. And so what we can do is we can partition a 384 well plate into quadrants to allow us to study multiple variants in the same experiment. Now for KCNQ2 channels, we also have the advantage of a highly specific blocker known as XE991. And what we've routinely done is we've applied at the end of an experiment a saturating concentration of this compound to block all of the specific M current activity. And then what we're left with is some residual outward current that we attribute to some background channel function in CHO cells. And this can be subtracted digitally offline so that what we're left with is pure M current. Now, I've shown you how we've done this with a co-transfection strategy, with KCNQ2 and Q3. Since then, we've actually moved on to develop a stable cell line expressing KCNQ3. So now we only have to introduce the KCNQ2 variant alone, and that's led to a bit more consistent experiments. And I'll show you in a moment how we've harnessed that system to do co-transfections of the mutant allele with a wild-type allele to look for dominant negative activity. So knowing this, what I want to just illustrate for you in a very superficial way is our current efforts to study 80 KCNQ2 variants that are shown here graphically distributed across the protein. Many of these are disease-associated. Some of them are associated with the benign familial neonatal convulsion syndrome, and others, in fact, most of the disease-associated variants that we've studied are associated with the more severe early onset epileptic encephalopathy. You can see there's a large cluster of the variants in the S4 segment of the channel, but there really is a broad distribution here. And also, we've included several variants that were known to be pathogenic from prior studies in the literature using manual patch clamp, and we included those as controls. We also included about a dozen variants that were rare in the general population but were not associated with disease. These would be so-called population variants that we thought were important to give us an idea of what the background genetic burden in this channel was. And so just to illustrate a few example recordings here, here are a couple of variants that were studied early on. T274M is perhaps one of the most widely studied KCNQ2 variants associated with epileptic encephalopathy. And this phenylalanine deletion variant, I think, was a novel variant. Both of these variants illustrate complete loss of function of the channel compared to wild-type currents. And there were several other variants, all mostly clustered in the pore domain, that were similarly loss of function. This is just an example of some of those shown here on an IV plot. Interestingly, not all variants were loss of function or had simple loss of function traits. We actually have found several that cause shifts in the voltage dependence of activation, as illustrated in these. This should actually be normalized conductance versus voltage. But here in the middle here in black symbols is the wild type channel. And two of these variants caused a leftward shift in the activation curve, suggesting that these channels activate at more negative potentials than the wild type, consistent with a gain of function. Some others cause a rightward shift, which would be the opposite of a loss of function. Now, when you're dealing with a large amount of data, as Jen said, you have to develop.
We seem to be experiencing some technical difficulties with the audio. I think I will need to reconnect Al.
Can anyone hear me now? I've just reconnected.
Al, we can hear you.
I'm very sorry about that. I'm not sure what happened. I'm just going to go back and just say what I started saying a few minutes ago, with just to summarize this large amount of data in a visual display. What we've done is we've evaluated current density as a percentage of the wild type channel and plotted that on an axis whereby 100% means there's no difference between wild type. A less than 100% would be a loss of function, and more than 100% would be a gain of function. Similarly, for the voltage dependence of activation, we've looked at the absolute change or absolute difference or delta between wild type and the variant. And if there's no difference, we have a zero delta, which would be normal. Positive delta would be a loss of function, and a negative shift would be a gain of function. And so just to summarize here a large number of variants, one should be able to appreciate on the left that many of these variants have a profound loss of current density and would be considered loss of function, but some actually have increased or higher levels of current than the wild type channel. Similarly, there are some variants that show differences in voltage dependence of activation, either loss in the direction of loss or gain. So summarizing this is what we're actively working on now. Now, virtually every patient that has a KCNQ2 variant in epilepsy is heterozygous for that variant. And so we wanted to evaluate whether or not the variants might have a dominant negative effect. And so we developed this system whereby we have KCNQ3 stably expressed in CHO cells, and then we can co-transfect with high efficiency with the MAC-site electroporation, KCNQ2, a wild type, and a variant in different fluorescent backgrounds. And here's just, again, a snapshot of some of the data just to illustrate. Here we took the prominent loss of function variants and performed experiments with the variant alone and separately on the right, the variant plus wild type. And what should be obvious is that many of these profound loss of function variants are not rescued sufficiently well with co-expression of wild type, suggesting they have a dominant negative trait. But some of them actually are rescued, and so these would likely, we would predict these may be associated with less severe forms of the disease. When we performed these experiments, importantly, we were blinded to the phenotype and are now in the process of, after unblinding, trying to make correlations with genotype and phenotype. The last part of my talk is pharmacological evaluation of the channels and the channel variants. And KCNQ2 channels, or M-current, is the target of a class of drugs that activate or potentiate the channel. And the one FDA-approved drug, retigabine, also known as ezogabine, is been thought to perhaps be a precision medicine approach to treating KCNQ2 encephalopathy. So using the synchro patch, we can demonstrate that retigabine at a high concentration does potentiate the current density. The mechanism by which that happens is to affect the voltage dependence of activation and shifts in the conductance voltage curve to the left. We've checked now several variants. In fact, we've tested this on all 80 variants, but just to give you sort of a snapshot of a few that we studied in the beginning, I saw a chat that says I can't see the slide, so I've checked the voice, the boxing image mode. I hope the slide has come back.
Al, we can see the slide okay.
Great, thank you. This slide is illustrating the peak current and the delta V1/2 of a series of 5 variants that were our first test group for retigabine. And what should be pretty obvious is that not all the variants respond with respect to current density with retigabine. So rarely we see a variant that's actually inhibited by retigabine. So this is important to know, I think, for any pharmaceutical companies that are thinking about developing for clinical applications retigabine or second and third generation M-current potentiators, because it may be that there is some genotype-specific pharmacology inherent in using these drugs. That point is illustrated a little bit further here. This is a heat map of the response to retigabine among all 80 variants that we've studied to date. This is a series of 2 columns. The first column of each of these 4 sets is the percentage of current following application of retigabine as a percentage of the wild type channel. And then the second column to the right is the shift in the voltage dependence of activation in response to retigabine. And those that are colored in green are the strongest responders, and those in red are non-responders. These data were collected with the expression of variants in the KCNQ3 stable cell line in the absence of wild type. So we have separate data now showing that with the heterozygous condition, you see a different response pattern, but I didn't have the data to show you today. So anyway, just to summarize, among the monogenic epilepsies, probably about close to 40 or 50% of them have ion channels associated with them. Many of the variants detected clinically and also in research studies can be classified as variants of unknown significance. And we've been trying to use automated patch clamp to decipher and categorize variants of unknown significance by their functional attributes to try to improve the diagnostic quality of the genetic testing. And this is also useful for determining whether there are genotype-specific patterns in the pharmacological responses. And so just to acknowledge the many, many people who've helped with the study, of particular importance is Carlos Vanoye, who runs the SynchroPatch and is responsible for all the data I showed. Reshma, Sneha, and others listed here are our molecular biology and cell biology team. I didn't highlight any work that we do with our collaborators, Evangelos and Dina. But many of the variants that I showed you today come directly from an effort by Ed Cooper, Ingo Helbig, and Katie Helbig, who represent a variant prioritization core function of our NINDS-funded Center Without Walls, a grant that has supported this work. And I will also say that it's a privilege to have the opportunity to have Jen Pan as a collaborator in our center. And so with that, I'm sorry for the technical delay, but I think we're ready for questions. Thank you.
Great. Thank you, Al, for your presentation. Again, apologies to all our listeners for the technical difficulties that we experienced during Al's talk. We hope that you received all the information well, and we're happy to address any of your questions one-to-one as well, alongside making this transcribed for you so you can watch this in greater scope. So there were a couple of questions asked during Jen's presentation that I'd like to go through. A listener has asked, do you think your approach on Cas 3.3 can be translated to other GWA risk genes? Jen, are you still with us?
Oh no, we've lost her audio now.
Okay, apologies. So I think what I'll do now is I'll go through Al's question, and then maybe Jen will rejoin.
Can you hear me now?
There's a lot of echo.
Jen, we can hear you, but I think there's a lot of background noise and echo, so I think maybe perhaps there's a discrepancy with the connection. I think you're going to have to rejoin from the beginning.
Can you hear me
while
now?
you're doing that? Now it's better. Now it's better, Jen.
Okay.
Thank you.
Sorry about that. I'm sorry. Just there's some technical things here. So I wanted to take up that question. So it's a very good question, and it's something that I think it's discussed and debated a lot within Stanley Center at Broad, as you can imagine. Personally, I do believe that it could be a framework for dissecting some of the risks, molecular phenotypes of risks. It may not work for every gene, but I think ion channels, a particular class that could be useful because this is where electrophysiology can identify different dimensions of the property, which, as you can imagine, would be very difficult for proteins such as scaffold. You don't know which partner it is interacting with, and you don't know which aspect of the properties is more relevant. But for the electrophysiology, perhaps it is a little better situated to be dissected using this approach. But it may or may not work for every gene, even in the ion channel. It really depends on what is the potential associated molecular phenotype to the risk. If it's, on the properties that where electrophysiology can dissect, I think that would be the best scenario. But if the risk is, in the C-terminus and it's with the interacting partner, you may or may not be able to dissect it out using this methodology.
Okay, great, thank you. And then another question, how well does the noise analysis align with single channel recording in your head?
Okay, so we only have one example that we did. We published in 2016 of a single channel recording of this RH variant. So we did a noise analysis on that variant used for us as a validation. It aligns, almost better than we thought. So using analysis, we could see that it has half of the channel traffic to the surface, and the single channel conductance didn't change, and which is the case in our single channel recording. The conductance didn't seem to change, but the number of channels on the surface using biochemistry and whole cell current density is different. Now, having said that, it's the number of channels on the surface is better estimated than the conductance. So we can definitely, it depends on the initial curve we use the closing of the channel as to analyze. I wish I had more time to go into it because there's really a lot to be said about that. But in one variant that we see a perfect match.
Okay, and then one last question for you before I go into Al's questions. Regarding cell-to patch, one would imagine the transient transfections are not indicated. Have you made monocle stable cell lines for each of the variants?
Right. I chatted briefly about that question, but I'm glad you brought that up because in our approach, we did the single cDNA integration in a specific genomic location. This is a T-Rex system from Thermo Fisher. So each cell line, if you go through the system to constructing the cell line, is supposed to have only one cDNA integrated at a specific location, a specific genomic location. So it's rather homogeneous. It's not single colonies. But I think Al probably has more to say about this particular question because he has been using a lot of transient transfection to get at the function, and that even at the single colony cell line construction, we still see a lot of variability within a population. I think, as Al and I were just discussing this a week ago, there's a lot of biology go into it, and even with a single cell colony, you still see in our hand, we still see about 5 to 10% of the cells do not express channels, even with perfect seal. We think that might have something to do with cell cycle. Is something that we don't understand and we cannot control. But so far we have in my group we have used a single cDNA integration system to address this question.
Thank you. Maybe Al, you want to add some comments to this as well?
Yeah, I just echo that there is a lot of variability cell to cell. The advantage of the 384-well platform is we can collect data on 50 to 70 cells per variant without much trouble. And I think that provides us a lot of statistical power to discern true differences between wild type and mutant. And by the way, our experimental protocols, we always assay the wild type channel on the same plate as the variants. And that gives us a great one-to-one comparison. We're trying to eliminate as many technical variables as possible. And what we're left with is some biological variability cell to cell that's inherent in doing these experiments. And that's not unheard of. I mean, manual patch clamp, there's a lot of variability, some of which is eliminated by observer bias because an observer will pick a certain type of cell. And so we're beginning to come to the conclusion that it's better to be blinded to that attribute of the cell culture than to actually go in and cherry pick cells that look a certain way. That's a bias, and you eliminate that bias when you're blind. So, but I think the fact that we can collect so many replicates gives us a better sense of what the true difference between a wild type and a variant is.
Okay, great. Thank you for that. Some of the other questions, Al, what other type of ion channels have you studied using the synchro patch?
Yeah, so I focused on KCNQ2 today, but in fact, our prior work that we published a year and a half ago was on KCNQ1, a cardiac channel. And that's, again, a channel that requires obligatory presence of another subunit. In this case, it's not a channel-forming subunit. It's the auxiliary protein KCNE1. And we used a similar format, and that was very successful in studying about 80 variants in our first paper, and now we've studied another 40 or so, with more to go. We've also done work on now 3 different sodium channels, cardiac sodium channel and 2 brain sodium channels, as well as another brain potassium channel, KV 2.1. So all of that work has been done in heterologous cells. We've also had a lot of recent success recording sodium currents, potassium currents, and some calcium currents from iPS cell-derived cardiomyocytes. And that's something we're actively optimizing now.
Okay. We had a question that came in regarding myocytes as well. In another question that was asked for both you and Jen, have you done reverse put KCNQ3 allele variants into a stable WT KCNQ2 cell line?
Oh, yeah, so this is something we will be doing. We haven't done it yet, but ultimately we would like to study variants of unknown significance in KCNQ3. There are a lot fewer variants in KCNQ3 than KCNQ2, and hence we've focused on Q2 to date. But that experimental paradigm would be what we would do. So far, we have not made the stable KCNQ2 stable cell line yet, but that's on our drawing board.
Jen, do you have any comments to this question, perhaps? I'm not sure if Jen... Yeah. So let me go into the next question then. Al, someone has asked, do you feel that automated patch clamp findings need to be confirmed by manual patch clamp studies?
Yeah, in fact, we did that extensively and have continued to do that extensively, but we can't do that for every variant. It would defeat the purpose of the whole operation. But when we tackle a new channel, we pay a lot of attention to making sure that we include variants that have been studied extensively by manual patch clamp, whether it's by our lab or another lab, and to make sure that we get a concordant result with automated patch clamp. But to validate every variant with manual patch clamp is not practical. And I think we illustrate that very nicely in our 2018 paper on KCNQ1. We validated the assay up front by looking at 10 known variants plus wild type. And we saw very concordant results between manual and automated patch clamp. The exceptions were some variants published in the literature made some interpretations of the results that we wouldn't have made, but otherwise the results were concordant. We're doing something similar, but at a lower level with KCNQ2 at this point. But we've done it as well for other channels, and I think it's an important thing to do with assay validation in the beginning, but then we're very confident in the results we get with automated patch.
Yeah, I agree with Al. I just wanted to jump in there. I mean, I feel like if it's purely on about physical property, if you are confident that you have formed a seal and it's a high quality, passes all the QC that you set out to do, and then I think given that, the solution is slightly different, I think, with the automated versus the manual. But if you're focusing about physical properties, I think as long as you're confident with the QC parameters, at least in the few variants that we did and with Al's and in his experience, we're quite confident of the result.
And then as a final question, again, addressed to both, have either of you tried to record from iPSCs?
Well, I mentioned briefly, we've done quite a bit of work with iPS cell-derived cardiomyocytes, and that's gone very well. In fact, we have a paper under review now where we did an extensive pharmacological study of sodium channel blockers in cardiomyocytes, where we looked at 5 different drugs, full dose response curves, probably over 1,000 measurements. That would have been absolutely impossible to do with manual patch clamp. All voltage clamp, by the way. We don't have a current clamp amplifier in our system, which was one of the first systems, I think, off the assembly line. But the voltage clamp has worked very well. The efficiency is not as high as heterologous cells, but it's sufficiently high that we can do targeted pharmacology on cardiomyocytes. We have not done iPS cell-derived neurons, by the way. It's something we have thought considerably about, and we do use iPS cell-derived neuron technology in my lab. The challenge is that we grow our neurons on a bed of primary glial cells, which when you suspend the cells or disperse the cells for application to the syncropatch plate, the glial cells will likely be in sufficient abundance to basically, we will be sealing on glial cells more than neurons. So we have to come up with a strategy to separate the neurons in the glia.
Yeah, this is Jen. I just wanted to jump in to concur Elle's point that most of these iPSC-induced neuron protocols include glia cell, and the way we get around this, we didn't try extensively, but we tried a couple of times, is use a marker to do flow cytometry. So you harvest the cell and flow cytometry to isolate neurons and then patch. One of the things that we discovered, actually, because neurons has a lot of processes, you really need to work these neurons very extensively before you put them on the synchro. Otherwise, the processes may block the aperture. And then for the whole cell current, density and kinetics is great. The efficiency is low as you would expect, but you can record potassium, sodium conductance, positive and negative conductances. But they're not very stable in our hand. We haven't tried extensively. We tried a couple of times. We were able to record, but we have not really done any functional studies with them.
Thank you, Jen. And with that, I think I will conclude the Q&A series. Any questions that have gone unanswered, we will then include in the transcribed copy. Additionally, we will provide the on-demand video so that you can continue to watch the presentation at your own discretion. Again, I would personally like to thank Jen and Al for joining us today and giving us a glimpse into their research. Again, we really do apologize for any of the technical difficulties that we may have experienced throughout the presentation. We look forward to having you join one of our future sessions, and we welcome any additional questions directly to us and to the attention of our presenters. We wish you a great rest of your day.